All right, part two. I wasn't keeping an eye on the time, so that was just randomly cut off. But yeah, I think I was talking about collision theory. So basically, the idea that a substrate and enzyme have to collide at the right orientation, facing each other, and with enough energy, so kinetic energy or moving energy, to be successful. And when you link all of that information together to truly answer the question, which is discussing why it's important that there's two different temperatures in the process of making this bread, um, we can kind of say some of the following things. The optimum temperature is the temperature at which substrates and enzymes have the most kinetic energy and collide frequently, but not high enough that they denature. The rate of reaction or enzyme activity is highest at the optimum temperature, so that's what we want. All enzymes are different. Here's the key. We're comparing and contrasting the different enzymes at the different temperatures to talk about how they're different. They have different optimums, and what's happening to each of them at each optimum. All enzymes are different, and they have different optimums for each factor that affects them. We can see here that the enzymes in the yeast that are responsible for cell division and fermentation have different optimum temperatures. So they need different steps in the baking process to be at different temperatures for everything to go smoothly. At 26 degrees, this is my compare and contrast, the yeast cells are dividing quickly due to the enzymes that are involved in that reaction being at their optimum. But the chemical reaction of fermentation will be going very slowly as the enzymes involved in this reaction work best under warmer temperatures. At 35 degrees, the enzymes in the yeast that are involved in the fermentation reaction are working at their best. However, this temperature surpasses the optimum temperature for the other enzymes, the ones that are involved in cell division. So their activity will be slowing down as they start becoming affected by the heat. So that's the excellence level comprehension of kind of what's going on in this process to the different enzymes involved in different things. It is important in bold there to recognize that we're not talking about the yeast doing these things. We're talking about the enzymes that live inside the yeast cells carrying out these processes. So if you talked about the yeast is doing this, the yeast is doing that, that can only get you to A or M because you haven't highlighted the fact that you understand it's the enzymes inside the yeast cells carrying out these processes or catalyzing these processes, not the yeast itself. Continuing down to question D. If the concentration of sugars is more than 6% of the flour, so if we increase the concentration of sugar in the mixture, the rate of fermentation is slowed because there is less water inside the yeast cell. So that's giving you a hint as to what you need to talk about. Explain how the increase in concentration of sugar will slow the rate of fermentation in the yeast cell. You may use a diagram. So basically, we would expect that as we increase the amount of sugar, that more fermentation can be carried out to turn that sugar into CO2. But this question is saying, despite what you think, actually, if you increase the sugar too much, then fermentation slows down. And it's got something to do with water. What do you think that is? So the clue for this question was you will be talking about a type of cell transport. Again, the word water was highlighted here. It's got something to do with water. So here's the model answer. If there is a high concentration of solutes outside the yeast cell, in this case we're talking about lots of sugar, then water is going to be moving from inside the yeast cell to outside into the bread mixture with the sugar in an effort to balance the concentration of water and solutes in both areas to reach equilibrium. That is our definition for which cell process. It is osmosis, the diffusion of water across a membrane, semi-permeable membrane. This process is called osmosis, and it's a passive process which will occur in the presence of any water solute gradient, any differential between the concentrations of water in two areas or solute in two areas, and osmosis will occur by itself. It just does it. Although the enzymes in the yeast will become more active as sugar concentration increases initially, there will be a limit due to another interrupting factor, and that is water. So we know that all cells, including yeast cells, so all cells of life, are made of mostly water. So the cytoplasm, the jelly stuff inside a cell, is pretty much mostly water with lots of dissolved chemicals and hormones and things like that. And so losing a lot of water will affect the cell's essential processes, because in the cytoplasm, that's where everything takes place. Well, most things. We know we have other organelles for lots of processes as well. But as the cytoplasm in the cell starts to shrivel as it's losing water because you've increased the sugar outside the cell, the cell's going to struggle to function and survive. As the yeast cell loses water, the rate of chemical reactions within the cell will decrease. That's all chemical reactions, including those involved in fermentation, cell division, etc. So the idea is, as you increase sugar, great, the yeast loves it for a bit, and then actually that sugar starts to affect the osmotic pressure in the cell, and it starts losing water. When a cell loses enough water, it can't do anything. So that's really the idea that it was hinting at, was that you could recognize that it had something to do with water, and it's something to do with increasing the amount of sugar. And so what could you connect there? The idea that the cells are going to start shriveling up due to the process of osmosis taking place. All right, E was completely random in my opinion. It has nothing to do with the bread yeast situation, but it is related to enzymes. So I guess the examiner is just trying to get as much enzyme knowledge out of you as possible. Heavy metals such as mercury and lead are enzyme inhibitors. E, explain how an enzyme inhibitor affects enzyme activity. You can use a diagram to help with your explanation. So, to break this down, describe what an inhibitor is and how it affects enzymes. Pretty much the question is pretty straightforward. An enzyme inhibitor, and this is another definition, you should be able to rattle these off. So great if you're using the glossaries on the iPad. An enzyme inhibitor is a molecule that binds to an enzyme to reduce its activity. And the only way that I could think to really expand on that idea was to talk about the two different ways that an enzyme inhibitor can work. Number one, inhibitors can be competitive, which means that they bind to the enzyme at the active site. So they're competing with the intended substrate for space in the active site. And you can see that on the left there. The yellow thing is the inhibitor, and it's taken up the active site, and now the red substrate can't bind. So it's stopping the enzyme from doing its job. It's inhibiting its activity. The other way that an inhibitor can work is non-competitively. So it's not competing for the active site. It's actually binding to the enzyme somewhere else completely. But in doing so, it changes the shape of the active site on the other side of the enzyme, and it still doesn't allow the substrate to bind. So it's either competing for the active site or it's not. But either way, it affects the ability of the substrate to bind, forming that enzyme substrate complex and be catalyzed, for the reaction to be catalyzed. So that's the question. I hope you were able to tick off some stuff that you got right and add in some points that you didn't expect, and maybe you can see now from an examiner's point of view how you can break down a question into kind of what's needed. So focus on definitions and terminology wherever you see them, start with that, and then yeah, continue to kind of, if it's achieved, you're kind of stating, defining, describing. At merit, you're explaining how something works in more detail, and at excellence level, that requires you to kind of look at data, compare it, contrast it, justify your reasoning, all of that kind of thing. Yeah, I hope this helped, and I'll talk to you later. Bye.